offer connections, you offer and mentoring, you offer public science and outreach, thanks to Lauren, and there are connections with other clubs, with the Biotech Club and Sarah Kate. So you're doing a fantastic job in a time where it's difficult to have uh, research experiences in the lab. You have really added the creativity aspect that goes along with research, creative endeavors, and discovery. And what I love is you're yet another success story of how things came along and a group formed, student driven, and you were able to do some really neat stuff. I have a couple of slides to show if I could, if I can share my screen.
Yeah.
Who will let me?
All right. You should be able to now.
Okay, and I'll go quick because Osvaldo has already seen some of these. But what I wanted to do is, hopefully you can see this, and I'll go through the first couple quickly and tell you how I got to bacteria and Delphia, give you the story, and then hopefully convince you about something I want to do this spring. And I don't, well, I want to do it, but I want you all to do it. Okay, so let me see. Oh, one thing I was gonna do. Um, I can easily share this with everyone too, just in case. So you should have access, and I'm going to ask you some questions. But let's talk about a little about. I'll start with me, but then I want to get to what everyone has done, and talk about what several of you on this call have done. So I'm a faculty in at the Biotechnology Program. That's 100% of my teaching is there, and several of you are doing bit minors. And if you ever have questions or I can help in any way, do not hesitate to email, and we can schedule a time to talk about modules or talk about what research experience may be um, required and what counts and what doesn't count. And my home department is biological sciences, and I'm really proud that I'm a member of that department because I think biosci has really done a lot for trying to listen to our students and offer as much of a student-centered experience as possible and make sure that you are being treated fairly and there's equity and diversity. And I know we have a lot uh, to accomplish and there's things are not ideal, but I'm really grateful that in our program, in our department and in our program, we're talking about that. So a little about me. Go I'll go quickly because some of you have already seen this, but I grew up in this colonial town slash city. It actually looks uh, a lot like this nowadays with COVID, empty. But it's cobblestone streets, lots of expats that for some reason retired down there, and it was just a fun place to grow up. And my mom is an elementary school teacher. My dad retired from the US Navy, and I was exposed to a whole bunch of really interesting people like Ruth and Conrad Blumpe that came from Germany and really helped me get into science. So I come from this city in the middle of Mexico. And once in a while, when we're able to travel, I'm able to go back with the family. But it was a really artsy place, so I quickly went one state over to do something called a technical or science high school. And in Mexico, the science, many times high school systems are attached, especially private ones, are attached to universities. And I was lucky to get a scholarship to this program that was kind of an experiment. It was bicultural high school. So I had English classes, like math and English, and then science and Spanish. And then the next semester they would switch. Several of us struggled along with that, but I grew up speaking both Spanish and English at home, so I loved this, and it really prepared me for my next step. But in high school, I met Gabriela Corral and Janet Simmons. So Janet Simmons took me as a grader, and part of my scholarship was grading math papers and math homework for in throughout high school. And Gabriela Corral was a biologist and is really interested in teaching. And she designed this class called Scientific Discovery. And it was going through the scientific method. And I did a project on biotechnology and Ebola viruses. And I was hooked. So they were convinced that I was going to be a physicist at the Tec de Monterrey, and I loved the Tec de Monterrey, but I was like, no way I'm going to be a physicist. And I'm stubborn. And I said, I'm going to apply to only places that have biotechnology, mostly to annoy my guidance counselor and mostly to convince them that biotechnology was something that you could major in. So I ended up applying to a whole bunch of places and for some odd reason ended up at Worcester Polytechnic Institute in Massachusetts. So from sunny 70-degree San Miguel Allende, I went to this awesome place, but very cold. And there, a fantastic biochemist from Argentina took me to do undergrad research. And I'm missing one name there. Jeff Tyler was a population fish modeling person. So both Jose Arguello and Jeff took me along to do undergrad research. So I would go from one building doing feeding fish as my undergrad research job and measuring how long fish could swim in PVC slash acrylic tank setup we had where water would flow and we would measure fish bioenergetics and then we would model them on the computer. And I I thought that was a kind of weird, but also fun and relaxing. And I love the coding and I love the computer part. And I was like, I'll learn some coding and see what happens. And then with Jose Arguello, I cloned my first gene, learned about bacteria, and turns out that the two things, the two experiences I did really tied together to my next step, which would be doing a PhD in microbiology because I like the microbiology and I also like the computer and the automation. And this person over here, Dawn Johnson, was the director of Minority Affairs, and she really created a cohort, a community where we had peers we could um, work with. We had study groups, and thanks to her, we were able to really get the most out of the WPI experience. And then I went to Emory, and I did a PhD with Tony Romeo studying bacteria and how they attach to weird surfaces. And uh, and we focused on E. coli, and I really enjoyed it. I learned some bacterial genetics. I learned classical genetics, and doing some really neat, like using bacteriophage to move genes in and out of E. coli, and doing biofilm assays. And he really let me do two things that were really cool. Most of his lab worked on RNA binding proteins, and I know some of you have worked on RNA and things. And I was one of very few in his lab that was not on the RNA side. I was stuck on, pun intended, biofilms, how bacteria attach to things. And he really let me go after this project and find what regulators, think about bit 410 and or genetics 311 and 312, promoters, and what transcription or regulators affect regulation of the genes in E. coli to make polysaccharide. And the other thing he let me do was take classes in the School of Public Health and teach. So Tony signed off every semester for, dare I say, 6.5 years. Yeah, that's what it took me. 6.5 years for me to take classes. So even when I was done with my required classes, he let me take classes in the School of Public Health, and he let me TA with two really awesome teachers, Pat Marsteller and Ari Eisen. And they were really great science and public science mentors, and also on the teaching side, how to really encourage students to be active in the classroom. And then I went to Duke, and I did more of the clinical side with Pat Seed, looked at how bacteria um, are important in the clinical setting and how biofilm formation happens in bladder cells. So Pat, MD, PhD, fantastic mentor, but also really creative. And I remember like taking apart a printer and duct taping pieces together to try to spot bacteria on plates, doing high throughput assays by hand, trading off a multi-channel pipetter. We did 2,000 samples or 2,000 different compounds by doing multi-channels and trading them off. So he was fantastic, and he is fantastic, and he's now at Northwestern. And Doug James allowed me to learn about teaching, and I also met my better half downstairs, who taught me how to inject mice at Duke, and allowed me to learn how to do mouse experiments without injuring myself. And then I came to um, NC State to learn about teaching. And I'm grateful to lots and lots of people. And I have the library here because the library folks have been fantastic partners. And they have really helped me connect the two things that throughout my career have really been things that I find really rewarding, which is teaching and science and public science. And the library has been that cohesive factor that has let me put two and two together. So I tell you this because I learned that mentors are essential. So throughout every stage, I had a mentor. I had a community, either a student group like yours, or a, a cohort, like grad school cohort, or at Duke, we were part of a cohort of scholars learning about genetics and um, bacterial pathogenesis and the clinical setting, so interacting with clinicians and connecting back to these people. So even now, I just sent a student to do email Pat Seed to see if they can work on a project remotely. And I often email Pat Marsteller from Emory, and now she's actually on the steering committee for a project we do. So these are mentors and allies that you'll have throughout your career. And I cannot emphasize enough that the people you meet, uh, keep in touch with them, not only for support, but also for ideas and for the energy when you need it. And follow your interest and curiosity. I was interested in bacteria and how they attach to surfaces. I never thought that feeding fish as an undergrad would pay off in some of the scripts I used later on in grad school. And the cloning that I learned with Jose Arguello and the protein assays to this day are really essential And some of the knowledge I hopefully share with students that take Bit 410 comes from some of the knowledge that I learned from my experience in running protein gels with Arguello's lab. So follow your interest and curiosity and learn. And now I have the opportunity to share that with you in the biotechnology program. And we have tons of techniques that we can now use for genetic engineering, for learning about the microbes and the cool things around us, and constantly changing field of biotechnology. Lots of connections to other disciplines and exciting opportunities. So if you go to our biotech page, we now have a news section. And you'll notice who's writing some of these uh, blog posts. Uh, Sarah Kate Connor from the Biotechnology Club. And I love, and she's being mentored by Dr. Stephanie Chen, but I love how we have the student voice in our program. We have minors, and several of you are doing the minors. So if you ever need help, here's where to find the information about the minors. And I'm just an email away, so just email me. And with this came a really interesting thing. I came here to teach a class about metagenomics, the microbes and their genomes that are around us. And it evolved into a series of projects. I later on, with Dr. Stephanie Chen, co-designed a high throughput discovery class where we learn about automation and liquid handlers and screens and high throughput sequencing and microfluidics. With my better half, we teach yeast metabolic engineering, and Dr. Claire Gordy and I are teaching it for the first time virtually this year, and having fun doing it with a fantastic group of students and learning how to best get you excited and working with yeast and learning some of the techniques. And microbiology, I always go back to it because of the metagenomics class I came here to design and my background. And pulling all this together, I want to design a 200 level biotechnology and sustainability class where we use some of the skills and some of the knowledge we have about the microbes around us to try to answer questions like, how can we recycle electronic waste? And I know Lauren and Rabea and several of the group members have worked on the Delphia project to really do research on the electronic waste recycling. But what I really like is co-creating. So this is our new Bit Metagenomics website from last semester. So you are well aware that you work in Moodle, right? For several classes and you have your assignments on Moodle. I want to go beyond that. So not just a paper that I read and it gets archived away, but I want you to produce something that we can use later on. So students in the class, and now I'm trying to do it for all our classes, create podcasts or tutorials or things that can help other people beyond our class. And I'm honestly inspired by uh, the, our Delphia group and open science and our libraries in making science accessible for everyone so that we can co-create and learn together. So what does that mean for us? At the Delphia project, and Deja Norman and um, uh, Noah Riley and several students um, uh, have really helped us learn about this microbe we found the first semester I taught the Bit Metagenomics class. We were swabbing kitchen sinks, and a grad student's kitchen sink had 60% of this microbe I didn't know about. And we Googled the name and Delphia appeared. And I was like, what's that? And I remember doing it on the screen and learning about Delphia and getting really excited because it had just been described that it has a series of genes to make a series of enzymes that are basically a conveyor belt of stringing together peptides to make delphtibactin, a peptide that allows us to precipitate gold out of solution. And I was intrigued. And it was really great because students were intrigued and you have really great questions. And together, our curiosity led to Victoria McQuaid and Vicky Cornish swabbing uh, water fountains, John Hip looking at whether Delphia grows in contact lens solutions as a BitSure student one summer, and several different projects. And the one you see on the screen is a Lauren Ramillo revamp of our really ugly old Delphia page. And what Lauren and Rabea and Deja managed to do with several of you was create this, make it an interactive place where we have a running blog where you get to share how we are engaging students in annotating papers, learning about Delphia. You are the researcher, and we ended up editing Wikipedia. So this is our dashboard, and these are all the edits, student editors, words added, uh, and article views. And I think that is really amazing that the group did this after not having lab access and looking for research opportunities. Lauren and Rabea and everyone else partnered together and just came up with what is a creative research project that doesn't involve a single bacterial culture. Although I went in one weekend and got some bacterial DNA to sequence, but you created and shared knowledge, and I think that's really important because all the student contributors really did it to learn more, and now we're trying to put that together in the form of a eventually peer-reviewed lit review on why Delphia is cool and why we want to put together all the information we know about Delphia that's in, as Laura and then as several of you have highlighted, it's very disparate or very disconnected. And what you're doing is really systematically using hypothesis and that nice number of tags that you've come up with. You've collected all this information. So you've not only impressed the Wikipedia people who just emailed today sharing the dashboard, but you've also impressed the hypothesis people saying, this is really cool. This is a use that we had not considered. Hypothesis for citizen science or student-driven co-annotation. So that's kind of where I fall in. I'm just interested in the gold pooping bacterium. But I want student-led projects, and I want this notion of we're a community of scholars. Let's learn more about something interesting and let's make exciting connections. And my job is basically to convince others that Delphia is worth thinking about by making dramatic videos, convincing people that uh, um, kitchen sinks that had Delphia, we should be asking ourselves, why is Delphia found in kitchen sinks? Learning that Delphia has been found in soil. What is it doing in soil? Is it precipitating gold? Or as Rose Krebs, several of you highlighted, including Lauren, maybe it's finding iron and trying to precipitate iron out with this delphibactin. A couple of you have identified papers that show Delphia acetoborans can affect plant growth in a good way. So it's a growth promoting bacterium. And if you look at patents, If you look at patents, a Canadian company, biotech company, actually has a patent for using Delphia to enhance growth. So it's a bio that it has a name that I'm forgetting now, but it's basically something that we could add to soil to enhance it, enhance its productivity of crops. And that's really interesting because there's on our campus the Plant Science Initiative with a big building coming up in on Centennial Park. And now I'm slowly convincing people, thanks to your research and help, that maybe we should look into Delphia for plant growth promoting um, properties. So the blog for me was a highlight, a success from all of you, because it really showed how you could organize yourselves, how you could share in a way that I have yet to understand how the readability score works, but you've made it accessible for lots of people, and you've made it publicly available. And again, that goes back to the community of scholars. So I've been thinking about this for a while, and I actually have the link if you want it. And Claire empowers me by making weird Bitmojis for me upon request. But I've decided that my research philosophy is a research mentoring philosophy, where I think that we should be a team. So if I mentor undergraduates, we are a team. We'll both learn. Science is collaborative. You bring expertise and ideas. Our diverse backgrounds and experience really enrich the brainstorming. And I I want us to come up with a project together. I want us to be curious. And I did not know about, um, for example, contact lens solution and Delphia and that connection. And John Hip actually did experiments before someone else published a paper. Um, so your curiosity really fuels what we focus on. And we will try again. K-Base didn't work the first time. Let's make it work. The robot dumped Delphia all over the deck. We'll make it work, but I'll let you struggle a little bit and then I'll seek help. And my job as an educator is I want you to learn. I want you to develop and grow. So we can always ask for help. And I'm learning, but we should be flexible. Sometimes I'm like, this semester, Rabea, we are doing 2 case studies and we are doing a I'm stuck on the podcast and we're doing a podcast and I'm convincing people that we're going to submit this sustainability grant that I'll get to. Um, and sometimes we can't because we're all taking classes, including Carlos, or teaching classes, and we have to be flexible. And sometimes that flexibility is not because of barriers, it's because you found something really cool. You found this cool wiki edu way of us collaborating. So with that, what now? So this is kind of a hodgepodge of weird graphics that I've made with help from lots of students. And they are all clickable because they are links to things. So hidden on our Delphia website, I mentioned, we have what we call the Delphia book, a book about the golden microbe, which we convinced the library to pay for a Pressbooks book, which is an open educational resource. It's like WordPress, but looks like a book. Lauren reminded me today about it and how we have some factually incorrect information that we have to correct. So that's an opportunity to, as we're working on our lit review and doing a research paper and editing Wikipedia, can we add more chapters to this book so that we eventually publish it and share it? And we have Will Cross and Micah Vandergrift in the libraries that have promised me that if there's something that has a cover and several chapters, they will help us publish it. Metagenomics, I'm always grounded in metagenomics because that was my first bit module to design. And that's how we found Delphia sequences. So I'm really interested in sharing tools and learning about the microbial communities around us. And that could include Delphia or some other interesting microbes. And for that, we have high throughput approaches. And that's do not fear the robot. How can we use technology to do some cool stuff? And with the better half, I teach yeast metabolic engineering. And this semester, we are going to do some case studies for Gala, for this platform where we can make some really nice looking case studies. Student names go on them. We can teach people about interesting uses of biotechnology. And it's something you can share because I want you all to have something you can share when you apply for jobs, when you apply to grad school. And the last thing that we're working on, and I wanted to launch it this spring, but it didn't happen. For several years now, I've really been itching to teach a 200 level class. And I've affectionately named it Bit 295 because it's a special topics, BS, biotechnology and sustainability. And that is merging electronic waste that several of you have really made me think about how we can use bacteria to recycle and reuse electronic waste and biotechnology. And for that, I think we have lots of opportunities for your research to tie into maybe creating case studies. Tanasha and Rabea have this Office of Undergraduate Research a grant to learn about the electronic waste recycling at NC State, in the state, and in the world. And here's my plug. You knew this was coming. You pay student fees, and some of your student fees go to the sustainability fund. And it's a grant option to request funds for projects that have to sustainability. So my challenge for you is, what do you want to do that they can fund that would impact students, that would impact our community, and that would be something cool to do? Because as my boss says, you want to be sure you're interested in doing it so that you are curious and motivated and want to learn more. So the applications are not due till March 12th, and it's kind of a form, but it's almost like a grant, and it will be a good experience. And what do you want to do next that combines sustainability and maybe biotechnology? So with that, my new one is sometimes you win and sometimes you learn. Sometimes it doesn't work out the way you intended, but I think most of the time we learn something new. And my role as an educator and mentor is I've come to realize maybe not just encourage you to um, learn, but really give you the confidence and the resources to try what you're really interested in. So with that, what do you want to try and how can we help you? We know there are limited undergraduate research experiences available right now. BitSure, NC State BitSure is taking applications, and we're going to do it either virtually or in person or half and half. Um, so I encourage you to apply. And what would you like to do next is my question for you. So what questions do you have for me? And how is SACNAS going to be involved in this and the Biotech Club and STEM Journal Club? And
Well, since you brought it up, we actually have, for those of you who don't know me, my name is Osvaldo Rodriguez. I'm the president of SACNUS chapter at NC State. And tomorrow we are having a general body meeting at 6:30. So if anyone's interested, you could reach out to Dr. Goller, as he has the invitations as well, or you can reach out directly to me. I'll put my email in the chat. But long story short, yeah, the Delphia project is one that's like very, very interesting to me because this whole time, well, until I learned about the Delphia project, I think mid last year, I thought that science had to be done within the laboratory or it had to be this thing that needed to be done underneath professors and just doing actual like, bench work and whatnot to get it. And here you're showing us like science continues going, even when we're just proofreading paper, reviewing papers and connecting the dots. And as I've started getting into more and more advanced work with biotechnologies and even within my own research project, I'm starting to realize that too with yeast. There's a gene that I'm working with to create mutants of this gene, the Mhr1 gene. And I just keep finding all sorts of different papers sort of disconnected from different disciplines across the nation, across the world about this gene. And so all the papers sort of say that there's not too much known about how the gene works. We just know it has something to do with the mitochondria and it could result in a petite yeast or not. But then they all have little bits of information that they discover about it. Like, oh, well, in this particular strain of yeast, when we mutated knocked down the MHR1, it did this thing and it did this thing and it correlates with this issue here. So I'm finding what Delphia is doing is really fascinating. It's sort of like bringing the importance of that open research and being able to connect those papers together. So with SACNUS, what I'm hoping to do is draw more students into this research opportunity by promoting it as much as possible, but also doing a blog takeover. So we have, and hear me out, this is for STEM Journal Club too. We have our own blog that we've recently started. It's called Las Letras, which also means the letters. And our whole intent on that is speaking about a variety of different wide topics related to inclusion of underrepresented minorities within STEM. And this includes cultural exchange pieces, pieces specifically about science, pieces about interviews with professors or notable people within our community, just to sort of like bring an entertainment sort of value and informative value to the expression of science. And so one thing that I think would be really fun is to do with all the other student organizations that have blogs, podcasts, or other content mediums to do exchanges in which they'll come to my blog or to Stackness's blog one day for 1 week, and they'll write content about our mission from their perspectives. And we will go to the other person's blog and we'll sort of exchange positions using our perspective to write not about what we want, but about what it is that they focus on and write it from the perspective of underrepresented minority scientists and people working on Delphia to write about like, well, this is why Delphia is important in all sciences, especially if we're talking about recruitment of URMs, that kind of stuff. So that is how I can see SACNUS sort of helping out in terms of getting the story across. And same thing with STEM Journal Club. I would love to do a swap with y'all. That would be a lot of fun. So I think the blog takeovers would be a fun thing.
I knew this was gonna happen. I really like the blog takeover idea. I think that would be cool. And I'm itching to write something for Las Letras.
Oh yeah, we took a hiatus last week and the week before that just to get prepared for the first week of school with the blog. But we are also working on a larger piece that has taken us some time. So I'll give everybody sort of a sneak peek. Our piece, because we don't just we're not just planning on doing just writings. We're also doing like mini documentaries and video based content we have been speaking with a nurse who's been working on a COVID ward, and she is a Wake Tech student who's been spending her life, just dedicating herself to trying to get into the nursing program. So we've got a really great perspective on what COVID has done, not just for her as a prospective nurse, but also as a student, and how it's, altered her career path. So
Now that that's quiet, that's awesome. I really like that because it's connecting real lived experience and different voices. And Myra Hack really did something phenomenal with this group by starting the blog, and well, the whole group did it. You all did, but I really love the idea of you posting recordings and summaries of presentations and things. There's an accessibility there because you can access it whenever, as well as documentation, and it really shows reflection. You've inspired me to start blogging.
yeah, well, I'm not gonna lie. STEM Club is like the thing that I've been measuring our success on. So like, I remember doing the interview with your founding members when we did it for the Highlight podcast, and that was really successful. Chris Powers and Woody, who are the owners of Triffy, that did the second follow-up interview for that podcast. They absolutely loved listening to students talking about starting up the STEM Journal Club. So you guys keep up the good work.
Yeah, thanks, man. It was really dope doing that podcast too. And the stuff you guys are doing, like obviously it's great what you guys are doing.
Thank you.
So what would help you and help other groups? And I know there's requirements, right, for research, but what would be helpful for several groups and for your careers?
I think it's kind of based off a lot of what you had talked about during your presentation, in terms of, I guess, people providing mentorship and building that community and all that stuff. Like it sounds like super cliche and all of that, but it's like for real. You know, if you have
Mhm.
people around you that you're able to talk to pretty easily, about things that you're interested in terms of research, in terms of academics in general, I think things like that go a long way, to help people get where they want to go.
First of all, I don't know why you put community in quotation marks. That's my first complaint. But yeah, I think starting the mentorship program was amazing in the winter. It really helped. I feel like a lot of people, even the mentors, I feel like it gave them experience on how to, because it's hard being it's hard being a mentee and it's hard being a mentor. So it's like, for me, it was really cool to see both sides because Lauren is an amazing mentor. She's here in the call as well, but she did an amazing job. She was always very nice and understanding and would always help me figure out stuff I never knew about. So it showed me like, this is what a good mentor should be like. And then on the other side, I hope I got to, be a good mentor to other people as well. So I think both aspects are very important, like having people, and I think another thing that STEM Journal Club, and I'm sure Osvaldo, your club does as well, is I think I learned this through Delphia and the clubs, but science, it's an important field to learn, but more than that, it's an important field to be able to teach other people because scientists or people in STEM fields in general, they master their own subject, but they forget that if you're talking to the general public or someone who doesn't really know what you're studying that well, then they won't be able to really understand what you're saying. And I think we could all see that with the COVID-19 pandemic, how, some of us might be educated about the subject, but it's very hard to, you know, teach the general public that scientific knowledge, especially with the vaccine, it's very difficult. So I think that's something all of our clubs and research try to accomplish is, you know, making the scientific literature more accessible to everyone.
And there's a science storytelling club with public science faculty, and I should share information about that. Bradley Alf is and they do really neat things. They do
I dropped the link to their blog. I was just emailing them today, so they've got that too.
Perfect, Laura. I missed their talk this afternoon.
Oh, it was wildlife photography. The guy was posting in like the Smithsonian and stuff, so he was really cool.
Yeah, we have some opportunities, even though we can't do lots of stuff to grow Delphia in the lab. I think I love the interactions between clubs, and I think the members from all clubs are learning. Several of you are members of multiple clubs, so it works.
Oh my God, I'm looking through their website right now, and I've been looking for contacts like that for a while now. I think I sent out, because you saw that email too, where I'm like, I'm having trouble like finding like people in communication and getting out of, you know, just like the immediate sphere of influence. I think that's a great start. I'm definitely going to be emailing Bradley there. Thank you for
Yeah,
sharing
he's
that.
fantastic. He's really cool.
Yeah, we're probably gonna be emailing him too. So just letting you know.
Just don't tell
we
him.
can make it work together.
Don't tell him. I sent you. I already asked him for a favor for this summer. So we are doing the NSF bit sure. We're doing it completely virtual. So I'm trying to do coordinate a series of workshops, and one of those workshops would be science storytelling. And we will make it open for bit people.
Oh, that's cool.
Yeah.
All right. Well, it's 8 o'clock, guys. So we're probably gonna go ahead and wrap this up. Dr. Goller, thank you so much for coming and speaking with us, providing us insight, on your background. I did not know you were like, bro, you've been everywhere. Like, I did not know all of that. So that in itself was pretty impressive. And then beyond that, all the stuff you guys are doing with Delphia, like obviously all of that stuff is super interesting and it's awesome work.
I'll thank you first of all, but that's you'll find out that in 6 years you'll say, "Oh, I've gone through all this," and you'll figure out how convoluted that all our paths are. And I'm really impressed with what you all are doing in different clubs. And the Delphia group is just an example of cool stuff we can do. And if anyone wants to participate, we can find projects at connections. And I the one thing I don't want to do is um, give someone a project or make them just work on annotating. I want you to have learned something from it and join Delphia. Learn something from it and get something that you will use. Like Lauren has to get a public science job somewhere because she's fantastic at this.
All right. Well, thank you so much, Dr. Goller. Thank you everybody else for coming. We're gonna be having our next STEM Journal Club meeting 2 weeks from now, 7 o'clock. We're gonna be having some of our mentees presenting, hopefully. So look out for the email on that. If you guys have any questions, anything you guys need help with, you can email us at stemjournalclubncsu@gmail.com, or rtahir@ncsu.edu. mscon3@ncsu.edu. There are a bunch of different email addresses. They're all at the bottom of the emails that we typically send. So you can email all of them if you want to. There you go. Hassan's dropping his email too. Yeah, but our next.
Zainab, you can go ahead and drop yours too if you want.
All right, well, guys, thank you so much. February 10th is going to be our next meeting, 7 p.m. Thanks everybody for coming. Bye.
Thank you.
Thank you.
And again, but if you ever need to email me or if I can help with anything, just Osvaldo knows. Rabea knows. Well,
It was
We
good to see
appreciate
all
that,
y'all
Dr. Goller.
again. Have a good night.
Yeah, thanks for coming,
likewise.
Osvaldo.
Bye
No
bye.
Bye.
Thank you, Dr. Goller.
My pleasure. Nice seeing everyone. And Gwen.